Breaking news. Cornell's founder and namesake, Ezra Cornell, was found murdered in the Arts Quad. Who could have committed such a crime? Based on eyewitness reports, we have limited our search to 3 suspects: Keggy the Keg from Dartmouth College, Rory the Lion from Columbia University, and John Harvard the Pilgrim from Harvard University. We have collected DNA from the crime scene, but before I pass it on to my scientist colleagues, I'd first like you to answer a poll everywhere question on who you think the real murderer is. Mark.
All right, so before we can find who actually killed Ezra Cornell, we have to understand what microsatellites DNA is. So microsatellite DNA are small sequences of genetic markers that consist of repeated units ranging from 2 to 6 base pairs of nucleotides. And they're used for human identification purposes because they do not code for a protein and are unique for each individual. And they're also called STRs, or short tandem repeats.
As you remember from what we did in the lab, the first step in analyzing DNA is DNA extraction. To solve this murder, we needed to use saliva samples to obtain the DNA of each mascot. Magnetic beads will be used to pull the DNA out of the solution, and 3 buffers will be used: a lysis buffer, wash buffer, and elution buffer. After following these steps, the liquid, which will contain the DNA, can be extracted for PCR.
Okay, so a method to amplify the specific DNA that we need to obtain is PCR, or polymerase chain reaction. And the ingredients needed are the DNA that we need to copy, primer, the nucleotides, and DNA polymerase. And once the DNA undergoes the 3 stages, as you can see on the PowerPoint, and multiple rounds of PCR, we will have enough DNA to use to identify who the murderer is.
After amplifying a DNA sequence with PCR, the DNA can be analyzed in several ways. One way is by using an electropherogram, which can be used to estimate random match probability and learn exact fragment size. The x-axis shows the number of base pairs, while the y-axis shows the relative amount of each molecule. You'll remember that the double peaks show heterozygous genes and the single peaks show homozygous genes.
So once we obtain the DNA, we could use gel electrophoresis to find out the fragment sizes. So the smaller fragments travel further in the gel, while the larger fragments remain closer to the well.
As you know, based on the Hardy-Weinberg principle, the possibility of 2 individuals having the same alleles at every loci tested is incredibly low. As a result, microsatellites can be used for DNA profiling. Many cases in the FBI use CODIS, the DNA database managed by the FBI, to identify crime scene DNA. We are going to compare the DNA from the crime scene to the DNA samples from the mascots using gel electrophoresis to find out who killed Ezra Cornell.
We have successfully obtained the results from our gel electrophoresis. Based on these results, who do you think committed the crime? Respond at polleverywhere.com. There's a consensus. It was found that the true criminal was John Harvard the Pilgrim from Harvard University. As we can see, the DNA bands on the gel electrophoresis for his DNA perfectly match the DNA that was found at the crime scene. What was his motive? He was planning to demoralize Cornell before the upcoming hockey game on December 6th. But thanks to biotechnology, this was the last upset Harvard will ever cause. Thank you.